Yes, sir. I'm audible, sir?
Yes, ma'am, proceed.
Okay, sir. A very good morning to one and all present here for our fourth section of webinar lecture series. It's my pleasure and duty to bid you all a genial welcome. I would like to express my sincere and warm welcome to our beloved President, Dr. M. Sir, who is the inventive person, guide the institution for the path of success. I would like to welcome the most dedicated, hardworking, delightful secretary, Dr. C.M. Vasavi, Madam. Without her, the event wouldn't be possible. It's a great pleasure for me to welcome our adored principal, Dr. M. Lakshman Swami, sir, who stimulate and motivate us to achieve our goals. And I warmly welcome Dean Academics, Dr. S.R. Vatan Shankar, sir. Welcome you, sir. I am glad to welcome our star of the today's section, Dr. Jagadish Chandra Bose Rajendran, who completed his bachelor and master degree in Dr. MGR University, Chennai, and also completed his PhD in bioengineering, in University, Seoul, South Korea, in collaboration with CHA Biocomplex, Pangyo, South Korea, one who owned 8 awards and also provided his knowledge about his scientific applications, and at present postdoctorate researcher in Mesoic Medical Research Institute, New York USA. I'm here with us to share his knowledge about cell cell-derived vesicles, isolation, functionalization, and biomedical applications. Welcome you, sir. It's my grateful to welcome Dr. Selvi Ma'am, Head and Associate Professor, Department of Biochemistry, who is a guiding confidant of our department. Welcome you, ma'am. I would warmly welcome the Organizing Secretary, Dr. K. Nirbhama, ma'am, Assistant Professor, Department of Biochemistry, and I warm welcome to Dr. R.T. Narendra Kannan, sir, Section Coordinator, Assistant Professor of Biochemistry. Both of them lead the webinar to the great exposure to the knowledge. I wholeheartedly welcome the faculty member, participants, alumni committee going to make the section to the great extent. Once again, on behalf of Biochemistry Department, I welcome you to all. Thank you for joining us. We look forward to all today. Thank you for joining us. Thank you, sir.
Thank you so much, Ms. Suganti. Now we call our one of our Dean, Dr. Madan Shankar, Dean Academics, to deliver his felicitation. He is a very knowledgeable and dynamic personality which we have in our institute. Now I welcome Dr. Madan Shankar to deliver a felicitation. Please.
Thank you, Dr. Narendran.
Yeah.
At the outset, a warm good morning to all gathered here for this valedictory and the final lecture session on the international webinar series, which is hosted by the Department of Biochemistry. This department is a very, very dynamic department. I'm very sure that the kind of activities and the kind of programs that the department is doing is actually giving an impetus to the students to turn their minds into structured research as well. So this program, at the outset, I'm also happy to welcome the speaker of this day, Dr. Jagadish Chandra Bose Rajendran from Masonic Medical Research Institute, New York, US. My wishes and congratulations to the most dynamic head of the department, Dr. Selvi, and the vibrant team who is actually doing a wonderful job under her stewardship. Now, biochemistry, as you all know, it's a very basic subject, and COVID-19 pandemic. There are a lot of inputs which have come from the Department of Biochemistry from different parts of the globe. For instance, when you see a particular diagnostic company called as a Blueberry Diagnostics, they have really come out with an artificial intelligence-based algorithm to identify the patients with COVID-19 and this AI-based tool that they have developed using markers like the blood lymphocyte count, the eosinophil count, the C-reactive protein count, the D-dimer count, and also the levels of ferritin. They are actually able to tell the exact precedence and exact incidence of COVID-19 in the patients with 100% sensitivity and also with 100% surety that the patient will be COVID-19 positive. Similarly, another algorithm is also being developed, which is actually useful for prognosis. So, the message that I want to convey to you all is that biochemistry, having a strong basics, strong foundation in biochemistry, you should also diverge into latest technological interventions like artificial intelligence, machine learning, and see how you can blend them to develop a tool, or you can develop some features which will be amenable or which will be useful for a regular day-to-day diagnosis or something like that. Similarly, there is also another study which is very interesting because I happened to come across these studies in about 2 days ago. A study which was done by the Boston University School of Medicine. They have actually engineered a human alveolar cell, which is a bioengineered human alveolar cell, and this bioengineered human alveolar cell is sophisticated tech, is actually put in conjunction with a sophisticated high precise mass spectrometry tool, and they are now able to identify several host proteins and the pathways that are responsible for the progression of SARS-CoV-2 virus in the lung cells. And one of the most important discoveries which this study has brought to limelight is that immediately after the infection of the virus, or immediately after the virus infects the cell, there is a massive madness in protein phosphorylation takes place inside the cell. So when they were able to compare a normal cell and a virus infected COVID-19 or SARS-CoV virus infected cell, they were able to see a massive difference or a massive change in the protein phosphorylation which is happening into the cell. Several sets of proteins were undergoing erroneous or not a normal level of protein phosphorylation. So, understanding this biochemical aspect of protein phosphorylation will actually help in throwing light, or will actually help in bringing new vistas and new methodology of understanding the progression of the disease, and also to deliver to, and also to discover newer tools for diagnosis and also for therapy. So, having said that, biochemistry has a bigger room at the bottom to do quality research and also to bring out novel means of therapy and novel means of diagnosis. And I'm sure such seminars and such webinars which are done today will actually bring insights into the minds of the students to think out of box and to think in a different perspective, so that they can come out with some new discoveries which will be helpful for the society. I congratulate the entire team of biochemistry for organizing this wonderful workshop, and also take pleasure in once again welcoming the speaker of the day, and wish the program a grand success. Thank you so much for the opportunity given, and thank you so much.
Thank you very much for Dr. Madan Shankar for kind words, and especially the highlights of biochemistry, and it will be definitely with the encouragement for our students to look up to their research and higher studies. Thank you so much for your spending your time, valuable time in this. Even today you have too many sessions. Thank you so much.
Thank you, Dr. Narayana. I may not be staying on the meeting because I've got another meeting at 11 o'clock.
Yeah, fine. Thank you so much. Yeah, thank you. So now we can move on to the next session. I wanted to introduce Dr. Jagadish Chandra Bose Rajendran. He is the postdoctoral researcher, biomedical and translation medicine, Masonic Medical Research Institute, New York, USA. The topic is cell-derived vesicles, isolation, functionalization, and biomedical applications. So this is mostly apt with our final year students, those who are studying cell biology, as well as other students or those who are going to study about cell biology. I hope the session will be more interesting. And now with this small note, I introduce Dr. Rajendran. I invite him to deliver his lecture. Over to Dr. Rajendran.
Yeah. Can you hear me, sir?
Yes, sir.
Yeah. Good morning, one and all. Myself, Bose, Jagadish Chandra Bose. Actually, I studied in KMCH College of Pharmacy. I did my master over there, and I roamed in and around Coimbatore. I did my earlier job in Coimbatore as a scientific executive manager. So I roamed the entire Coimbatore by bikes. Okay, that's a past story. Then I went to Korea, and then after I come to US. So Um, Today, I would like to present the research which we are doing, and almost nearly 5 years I have been doing the research with cell-derived vesicles. This is an alternative topic. It's an emerging area of research to the cell therapeutics. Even in India, the cell therapeutics are few people aware the advancement of cell therapeutics. This is beyond advancement of cell therapeutics, which is called cell-free therapeutics. So I'm very happy to share this presentation, cell-derived vesicles isolation, how we are going to isolate this cell-derived vesicles, and we can functionalize like targeting functionalizations and other functionalizations, like we can tag the fluorescence molecules on it, and we can use these vesicles for diverse biomedical applications, including like disease prognosis or diagnosis, in other area like therapeutics or drug delivery. Uh, can you able to see my second slide, sir? Just I want to
Yes, yes,
confirm.
yes, sir. Yes,
Yes,
sir.
yeah. In human body, there is lot of cells and different types. It's and one could say the total number of cells about 10 to the power of 14, which here they highlighted. This is I got it from internet source. It's not accurate one, but you can compare with other sources. Like human cells is like 10 to the power of 14 cells in total, and 84% of the cells are contributed by the erythrocytes, blood cells alone, and other than that the lymphocytes 1.6, and the 2.2 other cells. So you can see we have a diverse variety of different kinds of cells, including hepatocytes. The liver is the another bigger organs here, and platelets and erythrocytes. So I just want to show the outline. What is the cell-derived vesicles? First, we should know what is cells and different types of cells. Particularly, I would like to highlight the cells from the blood. For example, red blood cells. This in this field, the cell-free therapeutics area or the drug delivery area, most of researchers are aware about the red blood cells. Particularly, people's from the students from biochemistry, you are taking like red blood cells. Oh, we know that it's a blood, it's a component of blood. But I spent almost 10 years 10 years in my research career. Then last 6 months I spent to study about red blood cells. It was so amazing because the pathway, cell signaling pathway in red blood cells is completely different from all other cells, because these are non-nucleated cells, and this mechanism, its functioning mechanism is completely different from nucleated cells. Only red blood cells and platelets are non-nucleated cells in the blood, and platelets are completely different from red blood cells. So if you are curious about further, please read it. What is red blood cells, and what are the major protein components of red blood cells, and how it is functioning, like how it is conducting the oxygens from one area to other area, and how it is releasing this oxygens and binding the oxygens, those things. It's very interesting if you get a chance to read further. Okay, in blood there is red blood cells, white blood cells, and including platelets. There is a lot of cells are there, and each and every cell has unique surface proteomics. It's called, I would say, it's a proteolipids, means it's a composition of proteins as well as lipids, and obviously there is like sugar molecules attached on the surface. So each and every cell has a different surface. Based on these surface, the cellular property is completely different. Based on these properties, the cell migrations and cell recognitions to the foreign body, say for example bacteria, the red blood cells responds to the bacteria. Say for example the COVID-19 virus, red blood cells sense and responds to the COVID-19 virus. And plasmodium, the malaria parasites, the red blood cells are the first responders to this malaria parasites, and it is exposed to the blood. Similarly, white blood cells, the macrophages, the monocytes, these cells are the first defenders, or police cells, which functioning, protecting our body from other things. So these cells has unique proteins which act as a sensor to recognize either virus or bacteria or other foreign materials. So that's an interesting area of my major research, because I'm not focusing on like cell signaling pathway more and more in deeper, but I'm just covering on the surface of the cells, how it is, what it is made up of, either what kind of lipids it made up of, and what are the proteins are associated with, what is the major function of those proteins in the case of recognitions or bio material interactions. In the case of, for example, if we are injecting with some other chemical components, how it is interacting with red cells, mainly those proteins, how it is interacting with those chemicals. That's a major interesting area of my research. So I just want to emphasize the cell therapeutics. Most of people, particularly from biochemistry background or the clinical background, they aware about the cell therapeutics. Even though I just want to give a little bit background. Cell therapeutics are advanced therapeutics one, clinically available nowadays. It means it's approved like last 3 years and so on. Before that, you can see these 17 and 18 cells are approved by US FDA for clinical applications, mainly hematopoietic stem cells. Stem cells, the cell therapy area, stem cells are investigated almost a decade. Even I started my career earlier in the stem cell research, but after that I moved to that area because the clinical the clinical development of stem cells are very hard to reach. So people are struggling with that research. So hematopoietic stem cells and the recent success with the CAR T cells, I would say that T cells are the most advanced therapeutics available for the cancer therapy, and the highest therapeutic efficacy today is with CAR T cell therapies are reported. So these CAR T cell therapies are very, very expensive. Say, for example, it's a $400,000 per dose of CAR T cell therapy if you want. Actually, what is this? What we do is, say, for example, a person have a cancer, they apply T cell and they engineer the T cells to fight against cancer environment, then they get back to the patient. So to compare, this single dose of cells will cost around 2.5 crore in currency now, but a lot of biotech companies in and others reduce the cost, take the version of this. So this is the overall cell therapeutics. Next to therapy, a lot of other therapies, a lot of another one cells, the cancer and macrophages. All these are suggested and they are indicated for the therapy. And our top is self, how we are to be self-free therapy, this cell therapeutics, because these cells are every cell has own communication mechanism, brain and paramechanism to bias proteins, but a cell can respond and send the signal, send RNAs and DNAs to lost. And say, for example, the breast cancer patients, primary tumor is formed in some other area, and cells depart from there and brain. So it's sending signals, sending a messenger RNA and mRNAs and DNAs, all the materials to that secondary tumor via the packed vessels called, I would say that cell vesicles. People are called as extracellular vesicles. Collective cell vesicle we can classify. We can classify based their size, which is like 1 micrometer. People say 1 to 1 micrometer, and exosomes is in size vesicles. This is around 50 nanometer to 50 nanometer. Such as can say that it's a 50 to 300 nanometer in size. Another one interesting area of research, apoptotic bodies. When died, it's form of blubbing, and it releases apoptotic bodies contain a lot of information, DNA. So researchers, each and every researchers, they their own interest area. Like people, for example, I did a lot of on microvehicles. I'm not spend much more with exosomes. The reason is purification of exosomes are very difficult from the source. So in the microvehicles, it's banned and easily the purification is very easy. And I also did some work with apoptotic. That's still easy part because it's the size, so we can easily separate it in the whole solutions. So so I explained different kinds of derived vesicles. One is microvesicles and apoptotic bodies, and based on their size, just to classify that, people classify based on surface protein like CD63, CD81, and on, or some people classify based on their protein structures and on. I consider these are the cell. I compare all these are counter of synthetic liposomes. People from pharmaceutical background, they're aware about liposomes, which lipid bilayer structure. People, lot of researchers used to deliver the drugs using liposomes as a carrier. Instead of those carriers, my major research is I'm trying to stick derived vesicles as a drug delivery carrier system. So I consider these are the major points as advantages to synthetic one. These are biocompatibility. Vesicles has high biocompatibility because when you inject some foreign materials into the human body immediately start to react, which will produce a lot of immune reaction. But in the case of cell-derived vesicles, these are lipid in nature. Other than the proteins, it's less immunogenic compared to the synthetic version, and minimal toxicity because it's easily biodegradable. When it is entered into the body, it will be like 24 to 48 hours after the circulation. It goes to the liver or spleen there, disintegrated and eliminated. And ease of engineering, the surface can be easily engineered and we can make as a targeted exosomes or targeted microvesicles. Say for example, if I want to target some part of brain, I can make this vesicle to specifically target certain diseases. Brain tumor, some patients are suffered by brain tumor and there is no cure. There is only one drug available, that's called temozolomide. So case of these exosomes, now most of people are investigating the blood-brain barrier crossing ability of exosomes. So we can pack the drug into the exosomes and we can inject it. These exosomes can easily cross the blood-brain barrier and reach the brain tumor. So that's the packaging of diverse therapeutics and enhanced site-specific disease-specific delivery. That's the targeting delivery advantages with these vesicles. And these vesicles isolation, as I said in the topic, I'm going to discuss a little bit about isolation. There is so many methods of isolation. If someone interested in your lab, particularly, a lot of people are now, they have started working on this cell-derived vesicles. It's very easy procedure, but all we need to do is we should have a one ultracentrifuger, or at least we should have a one tabletop centrifuger itself. It's sufficient. If the tabletop centrifuger or epicentrifuger, that centrifugation speed is 14,000 or 12,000 rpm, that's sufficient to pellet microvesicles. In the case of exosomes, we need more sophisticated instruments. In the case of ultracentrifugations, because these are nanometer in size and so it's not a dense particle, so it's always float on the surface. So we need to do either density, sucrose based density gradient, or antibody coated agarose beads to capture these exosomes to purify. There is other methods like Sykes exclusion chromatography and acoustic separations, fractions and affinity chromatography, and so on. There is so many methods available. But if someone's are interested, I want to work on in this area, sir. If someone's are interested, I would definitely recommend people, those who are interested, please work on microvesicles. That's much more easier than this exosome part. And here just one diagrammatic explanation. For example, as I said, there is different kinds of cells. In the following slides, I will explain. But here, I just want to explain with one cell. It's called mesenchymal stem cells. Or you can take any which is in your lab culture in vitro. in your lab environment. When you feed cells with the media, particularly some cell line, most people probably aware about 293T, HK293T cells or any fibroblasts. You can feed the media with cells. These cells will physically in the media. It will release in the media. So you just collect the media, not the cell, and after that you centrifugate, do either and pellet at specific time. I can give those values also. Once you get the pellet, then you need to wash with a phosphate buffer and can use digest options. So you can deliver drugs to the, but it depends on the cell source. Though exosomes only reach, and not every cell secreted exosomes. It won't go to the brain. The origin from neural sources. Neural stem cells can easily migrate to the brain. Or cancer cells can migrate. The exosomes separated from cancer cells can migrate to the brain. Like certain cells reach to the heart. Most of cells goes to the lungs and liver, spleen, and so on. So as I said, this is one example, or you can find like dozens of methods to isolate any cell-derived vesicles, including microvesicles, exosomes, and apoptotic bodies. As I said, apoptotic bodies are larger in size. So when you do like 2,000 G-force, you can easily pellet apoptotic bodies. But in the cells, you do the 300 G, so you can pellet the cells, and after that you more like 2 G, you get the apoptotic bodies. Then the supernatant, take the supernatant and can go for like 100,000 G, where you can pellet the vesicles. From that you can fill the meat by the 0.100 nano filter. Then after centrifugate 4 hours at 100 to 200,000 G, where you can get a mixed exosome differences. If you want to purify exactly correct exosomes, then go for photography or photography. That's the only way to get a pure exosomes. Like microvesicles, you can get it here. Apoptotic bodies, you get it here, and you can do studies over this. So this is one example of my research. I published paper. What is I took murine breast cancer cell line 41. Then I cut the serum free media. So these starvations induce to release a lot of vesicles that extracellular vesicles, like that extracellular vesicles. And I put down cells, take the super, and then I pellet down place again for pellet, the derived things. In that loaded therapeutic. In the case of mRNA, the RNA is probably people are about the corona now by the Pfizer plus the Moderna. The company mRNA as a key molecule to get back. So this is a advanced therapeutic. So we did a fluorophore conjugate RNAs, low efficiency in the tumors. So we can analyze vesicles enough area can load the therapeutics. Like I want to somebody's want to load, which is a small chemical component. Anything you can load within that, or some about the acid, ARR, and so on. Loading is easier than the loading RNA, DNA. The reasons are like high lithium, the inner core is more hydrophilic. So if you want to load like hydrophilic drugs, it's easier. I did load. I got with vancomycin. It's a kind of hydrophilic material. In the case of mycin or hydrophobic minerals, it's to load these vesicles are very difficult. So to load DNA and those are as hydrophilic materials, so load such materials, people need electroporation method where they electricity to like a pore. By that way the DNA molecules get inside or just incubate molecules by passive diffusion or particular in between 18 to those did like the map by the the material. And other one for easiest study method, it's a free. When you see the lipids, fuse to thaw means when bring it to normal temperature, the lipids arrange as ribosome structure. By that entrap therapeutics in the nano kind of and the physical actions that in power like 0.2 nanometer filter the membrane you enhancing it in the vesicles. So physical, if somebody is loaded, they want to ensure that whether it's the vesicles are or we see vesicles by micro instruments or can quantify that within it. So there are a few quantifications are analytical tools I use this to these are expensive and I use in India, like EMP, other institutes they may have. So one is like confocal microscopy, particular super microscopy, we can see the exosomes. Macrovisuals, we can see regular microscope. Apoptotic bodies, you can see normal microscope, big size is larger, like meter in size. If you exactly the shape like lipid here, we should go electron microscope, then go for 20 nanometer you go per. And if you want to go for down, you should go for trans electron microscope. I have image of those things also. And this what it's called dynamic light scattering method, which is these were sold by Malvern. Uh, industry India have the work in anode have instruments measure size so you like if I measure my samples, I can get the size range like 100 nanometer or 200 nanometer and so on. And this is advanced instruments, it's called nanoparticle tracking analysis, so where we can see our particle flow through a camera, CCD camera. So by that way we can measure the concentration exactly like per ml of my sample, how many particles or how many exosomes are within that. So I did all these experiments and I have a sample data to show you. I will show later. It said early, we consider these are the, as I said, it's like lipid bilayer structure or multi-layer structure, which entrapped a lot of proteins come from cells. So these exosomes are extracellular vesicles. By nature itself, it has a lipids and proteins. So we can conjugate any fluorescence, any antibodies, or any peptides, targeting peptides with these proteins by chemical conjugation method, or we can use particular lipids like phosphatidylserine or DSP PEG, like pegylated molecule. We can use those things to target specific organs or specific cells like macrophages. So those things here they detailed. You can read it further in the paper because I don't want to go in deep in these aspects. There is few like microsites, you know, uh, companies are developing the tablets as potential drug carrier hole, like can tag the drug on erythrocyte Erythrocytes are in blood, so it can release over the period like 120. Don't need to take frequently, so it will enhance the drug circulation's time, as well as it will reduce the dose and toxicity also. This is another one example of upconversion nanoparticles. These nanoparticles are synthetic material. When you inject the human body, it will be recognized by macrophages or monocytes. So to avoid that, what they did is they buried within the red blood cells membrane. So they coated red blood cells membrane on this upconversions nanoparticles. So after that, it's not easily recognized by the macrophages, because the macrophages think that these particles are, it's like own cells. So another method, if you consider to engineer the exosomes, so we can tag like RGD peptides on the surface, and also tag fluorophore on where it is going within the body. There are many methods, chemical conjugation methods available to engineer exosomes or extracellular vesicles, even cells. So I don't want to go much more in aspect. With I limit those discussions. I just want to show what is my experience with this chemicals, but my own research. I want to present my own examples. Last 4 years, I have been working with cell-derived vesicles, and particularly blood cells. I'm running on the red blood cell one and and platelets. Similarly, in the past, I worked with stem cells, and now I'm working with pluripotent stem cells derived. And past, I had some work with adipose derived Korea, and in Stanford, I did work with stem cell vesicles and cancer cells also. As I show the slides, previous PA who's expert in cancer cell, particularly he's trying to find your triple negative scan. So by that way, I chance to work on this. So this is my story. After I did inform, came pharmacy. I did some years in industry where I worked mostly in family. Then to biochem in Korea for function to pursue my PhD in engineering in Chung University, Korea. From there, I did almost 2013, 2016, did my PhD there. Then after 2016, I moved to UT Dallas. Uh, I did microbubble ultrasound and so on. From there, I moved to the university where I worked on cancer cell biology to find a nanomaterial based system for the triple negative breast cancer and liver cancer and so on. I moved like a year before the Master's Research Institute, cardiovascular institute. Now they are established, and they are establishing themselves as a biggest biomedical research institute in this New York area. So I'm going to present my research slowly. So earlier time of investigations, but now you can find like 1,000 articles, cell membrane coated nanoparticles. You can find 1,000 articles. So these are the major advantages of coating cell membrane on nanoparticles on this nanoparticle surface. So these coating method can overcome the biological barriers. Particularly, it can circulate long time. Say, for example, if I use red blood cells to coat the particles, these nanoparticle can circulate long time, like 120 days in human blood. At least it can circulate like more than 30 days, which is very high compared to the currently available all the nanomedicines. And similarly, if I coat the cancer cell membrane, it can naturally go and bind to the tumor. So by that way, we can easily target the tumor cells. And similarly, we can, we engineer these cell membrane which directly target the macrophages. By that way, we can reduce the inflammation or we can enhance the inflammation. So we can tailor this disease therapy according to our convenient. This is, theoretically, it's a lot easier to say. In practice, it's very difficult to do those things, particularly engineering this part with cell. It's highly challenging one. I spend a lot of time on this. So another thing is, I also investigated cancer cells. For example, this investigating in cancer vaccine development and cancer drug delivery area. Of researchers showing us they isolate in August, and all that they the cancer cell membrane as a lipid like vesicles, and after coat the nanoparticle previously loaded with drug. So the major advantage is cancers cannot be recognized by macrophages as these has e mechanism. These cells also can recognize cancer cells. For example, cells, if that goes by the breast, if use the brain cancer cells, it goes to the brain. So we can tune, depends on the cancer material, so we can deliver the drugs to particular cancer. So as I said, I did initial research in Korea, so where I cultured adipose-derived stem cells. Adipose-derived stem cells are nothing but it's a fatty tissue. Every human has a fatty tissues in their abdomen and so on, and some other part. So we can isolate the fat from the healthy patients even, and we can separate the stem cells. From there we can, because these stem cells has unique properties, these cells can, it's a like hypoimmune property. It won't make any immune based reactions in the human body. So we can coat these cell derived materials on the nanoparticle surface, and we can deliver the drugs. So this hypothesis we tested in a severe hind limb ischemia model, in mice model. So what I did is I isolated adipose stem cells, and I peeled off the membrane, and then I coated on the nanoparticle surface. These nanoparticles are loaded with VGF, a protein, therapeutic protein. So you can see how I prepared. This is a transmission electron microscope of nanoparticles, and where you can see this the lipids on the surface, and this is uncoated particles, and this is a confocal microscopy where we make a lot of large size particles to show this images. These are cell-derived vesicles, and these are the nanomaterial one, and this is a coated thing. After that, we injected before mice, you can see. Then after injection, these most of particles goes to the liver, obviously, because liver has the biggest filtration system in our body, liver and spleen. So whatever it's exposed to the blood, it directly goes to the liver, where it's filtered and it's accumulated. So you can see a lot of things on day one. However, because of the stem cell membrane coating, you can see this is a injury area, means we intentionally cut this. We made a limb ischemia. So you can see here the particles slowly, like day 3, day 7, day 14, like you can see the particles slowly migrated to this area because of the unique protein. It's called CXCR4, which gives migrating capacity to the stem cells. So same way, it's has the accumulation disease stage. This is my earlier investigations. And after I applied the cancer cells, so when I moved to Stanford, I did a postdoc Professor Ramas Paul Murugan in Stanford School of Medicine Radiology. So where we did, like as I said earlier, we cultured 41 cancer cells, and then after uh, I induced to release the extracellular vesicles. You can see these tiny lipid layered vesicles which has a proteins of cancer on the surface. So these vesicles we coated on the gold iron nanoparticle, and we also tag the fluorophore indocyanine green to track where it is going. After that, we injected into the mice of previously established subcutaneous tumor model. Then we image with the fluorescence image, and we also some MRI imaging. So these are extra vesicles of RNAs and so this I fused by through this process. This is going material, and the vesicles through the membrane. This membrane get coated, and you can see this is the so you can see these are like small large vesicles cancer cells. So these vesicles carrying RNA and DNA and proteins to the other cells and converting the healthy cells to the cancer. That's happening in most of cancer, particularly in the breast cancer. It's obvious observation. So then we used these vesicles to coat the core is the darkest gold iron oxide material, and you can see the surfaces means the derived from extracellular vesicles. So here what I did is we make a hydrogel matrigel, and we added a lot of extracellular vesicles derived from tumor cells, and we cultured here a lot of tumor cells. You can see these hydrogels are a little bit flexible, which allow the migration of extracellular vesicles to the tumor cells. So you can see the video slowly, like slowly it will start migrating from the core to the cells. So you can see the intense migration of extracellular vesicles. So using those properties, what we hypothesize that if we inject into the mice model of cancer, so these particles, because of extracellular vesicle migrations, it will go into the tumor. So what we did is we first implanted a tumor subcutaneously. Then after that, we injected our cell membrane coated nanoparticle, means the tumor cell derived vesicles coated nanoparticle. So you can see this is like day 0. The tumors are established. Then we also monitor the tumor by bioluminescence imaging. I haven't showed those images. So first we confirm there is a sufficient amount of tumor within it. Then after we injected the nanoparticle, so as I said, after injection, the nanoparticle goes to the liver and spleen. Then the period, like 2 day, 6, slowly start to migrate to the tumor where it is. So this is a tumor. Yeah, see the particles are slowly comes to here at the day particles emitting from where it accumulated to the area. So these really therapeutics to reduce tumor size. We are this continually. We got promise. I'm working on that. We also did this thing, the MR. Um, Gold acid can see this because of that. In addition to that, I'm also associated with another project. As I said, apoptotic bodies are a little bit easy to work with. We populated apoptotic from different cancer cells, from breast cells, hepatocellular carcinoma, and glaucoma. It's a brain cancer cells. Then we use apoptosis. From that, we collect apoptotic. Then we load antibiotics into apoptotic vesicles to deliver those apoptotic to the macrophage, which is previously infected with the organism. Somewhat you can see here. What I did is I culture the after I induce process, it start disintegrating as microvesicle. I get those vesicles, then I load antibiotics. Say for example, people probably knows mostly penicillin or this is vancomycins, like so antibiotics can use. So we use vancomycin, and the same extrusions method we incorporated the drug. Then we use this as a therapeutic. You can see this is U87 glioblastoma brain cancer cells. So we induced apoptosis, and these cells released apoptotic vesicles. You can see these are apoptotic vesicles, and these vesicles are reconstruction process. We load the material that there. We test drug deliverability of these vesicles within the Staphylococcus aureus bacteria infected macrophages. So this is kind of experiment by Kyle is expert in this area. So what he is, he took macrophages as well as cancer cells. First, he infected with Staphylococcus aureus bacteria. So the Staphylococcus aureus bacteria special or the capability to survive in this macrophages. You can see these macrophages, the cells can survive. Here you can see things. So the green ones are, these are cancer cells, like big cells. You can see the green ones are live bacteria, which is surviving with macrophages. Then after, because of our drug delivery, we can able to kill those bacteria. The red ones are dead bacteria. So this experiments was performed by Dr. Micro Nagendran. He's also my friend and colleague. So here in this video, you can clearly see this is a macrophage. The green ones, you can see the yellow ones are dead bacteria because of our treatment. The green ones are the live bacteria. These are live bacteria. These are dead bacteria. The reason is there is no antibiotic can reach the intracellular of macrophages. At present, only vancomycin is available to kill this particular bacteria, Staphylococcus aureus, methicillin-resistant Staphylococcus aureus. Vancomycin is the only therapeutics available in the market. But these antibiotics has a poor penetration into the macrophages. Because of that, we used our strategy, like we can encapsulate into the apoptotic bodies. These apoptotic bodies can be easily recognized by the macrophages. Then it will be eaten by the macrophages, where it goes and release the drug and kill the bacteria. That's the whole hypothesis behind this. So you can see this is a macrophages which has infected bacteria, and because of our drug delivery, you can see this. All these are dead bacteria because of our treatment. And the same thing where this bacteria are located. This is a single macrophages. The whole blue color is doppy stained nucleus, and you can see this green colors are live bacteria. The red one, these red ones are the dead bacteria. I took a help of my friend. His name is Arsini, so he helped me to particularly visualize, because this is intracellular compartment of single cell where we can see the bacteria is live or dead. So he helped me, and another person is my Kumar. He's he has a lot of talent. So but this label RNAs, when we incorporated apoptotic bodies, then we injected into the mice. Like this is nude mice model. So this is healthy nude mice. We want to see where our apoptotic bodies generally goes. You can see it's mostly goes to the liver and spleen. This is ex vivo imaging. This is in vivo when the mice is live. So you can see this after injection, these vesicles immediately goes to the liver and it stays there almost day 4. The same thing we tested in, because these nude mice are less immunogenic, hypoimmunogenic. But in the case of blacks, B6 mice, these mice are, it's like more proper model to investigate this material biodistribution in human. So we injected into this mice, and see, compared to this mice, it's immediately taken by the spleen and liver, then after followed by it goes other organs. He also showed this particular biodistribution pattern by using mRNA based RT-PCR based quantification, and he showed this most of the delivered microRNA goes to the spleen and liver and then kidney and so on. We also did some molecular imaging. This is a photoacoustic imaging. We can see a lot of materials goes into the liver. So in additions, we also tested some other nanomaterials in pig model, like large animal injected nanomaterials into the vein of pig. After it goes to the, as I said, when we injected, it goes to the liver. Then we applied ultrasound to release the drug, and we indicated how much drug is released because of ultrasound effect. In the in study of Stanford, I went to research institute where I associated with Professor McCarthy, his boss. So we work in two aspects. One is material delivery, nano molecular. This is on city area of research, and it's very hard to work. A single particle can be used to deliver the for and can be used to disease, or we can monitor the disease therapy. For example, saying in theory, we are currently investigating a lot of these molecules, so I cannot disclose a lot of information regarding this. If you use a single nanotheranostick, a nanoparticle can go. For example, if I into the cancer, we can monitor over the period like a month what happened, whether cancer grow more, whether this cells are not. So most of information we get it using our nanomaterials. In addition, our materials are custom designed because these materials has a getting moiety use like antibiotics and so on. Specifically goes and buy T cells, not B cells. That's how more tailored to the patient. So this is beautiful example of ability. So this, whenever you materials, it's go that not he cleverly engineer the materials and image it. This is a heart, and using his molecular imaging probe, he can show that we can image the heart. For example, if somebody suffered by heart failure disease, we can monitor using our nanoprobes. These are currently in the testing stages, but we are aiming very big, so it's a lot of challenges associated with. So I would highlight, like, we use some polymers. Then these polymers are usually conjugated with fluorescent probe, where you can image and see where these materials are there. Then these also materials also conjugated with some antibodies or peptides, which used to specifically goes and bind to the disease cell. So we are planning bigger and bigger. So what we are using is, we can engineer the cells. Just before he told me that in university people are engineering. In our lab, we frequently engineer the cells. We not just engineer like regular cells, we also engineer induced pluripotent stem cells. In this lab, in the Masonic Medical Research Institute, people are working with induced pluripotent stem cells. And I know one of my Tamil friend, he is also doing from induced pluripotent stem cells to endothelial cells generations, and even he is working on the myocytes to generate a new heart from this induced pluripotent stem cells, and so on. So a lot of research is going on here. So using those technology available platforms here, we are doing, like, we plan to culture or isolate a lot of cell membranes and coat different materials from gold, iron oxide, polymer, and so on, and use our custom synthetic chemistry. We can tag the targeting molecule, and we can use diverse disease pathology. For example, curing a heart disease, targeting a heart, it's very difficult one. So we are aiming to now using these materials to target the heart for the therapy. So in additions, I have been investigating this because, as I said, I cultured the human-induced pluripotency stem cells, and we are isolating a lot of vesicles from these cells, and we process it. And after that, we purify nanovesicles. We are now loading the nanovesicles with drugs. We also coat these materials with this induced pluripotency stem cells materials, and we are delivering it. She's Professor Maria Condraitis. She's head of our institute, and he's my boss. So the peak of COVID, we are not just sat and not discussed like that company invented this vaccine and so on. We are the research people, so we come up with something like we should contribute to the society. So what we did is we discussed in our team meeting, okay, this virus, everyone's thought that this virus directly goes to the lung and it causes acute immune responses in lung because patients are died at the time, because these articles are published in June. But when we discussed these things are this year Jan or February, I think. So we started earlier to think about these virus directly goes to the heart and it causes like viral myocarditis. Because of that, the patients may die very quickly, because when the virus reaches to the heart, it's immediately making heart failure. And because of that, the patients immediately died or he passed away within 1 or 2 days. So we gave this this our opinion article. Before that, Boston University or some other universities, now we know a lot of lot of universities are now they are investigating whatever we hypothesize, because culturing this virus in our lab it's bit difficult. That's why we gave up this idea. But myself and professor, even my director, institute director, we a very earlier time we discussed and we hypothesized that whenever the virus is exposed to this lung, it directly goes to the heart, where it causes infections, and because of that heart failure, the patients is higher chances to passed away. The main reason for this hypothesis is this ACE2 inhibitor. Like entire world at the time, they are cried about ACE2 inhibitors. But my professor Jason McCarthy, he previously worked with this ACE2 receptors, angiotensin converting enzyme 2 receptors. We are used these receptors to target our nanomaterials to the heart. So the same receptors are used by virus to get to get into the heart. That's our hypothesis. So we published this as an editorial comment in Drug Discovery Today. Beyond that, I want to give some take-home messages. Like I discussed so many things like extracellular vesicles, exosomes, and so on, blah blah blah. But some students may not understand what is this. Please go to the clinicaltrials.government and what is exosomes. You can see, for example, somebody's come up with the idea like T-cell derived exosomes can be used for COVID-19, and they are testing in clinical trials now. You can see the clinical trial first posted on May 15, 2020, and they are now testing it. So these cell-derived vesicles, particularly exosomes and microvesicles, are so many people's, even a lot of Indian communities are working on this area. And it has an enormous potential, not just drug delivery or gene delivery. It also has a potential as a biomarker. We can easily isolate the exosomes from blood samples, urine samples, or salivas, and so on. And then we can, diagnose the disease, like the stages of cancer or the stages of other disorders. So at present, I just typed exosomes in clinical trial. It's a US FDA official clinical trial website. So there is more than 200 studies currently enrolled, and they are in investigation, these exosome-based materials. You can, if you have a time, just go to this website and check it, and it will be it will give some clue on this area. So, and I would like to thank my professor Jason McCarthy and my institute director, Professor Maria Condraitis, and my previous PI, Professor Ramasamy Palmurugan, and Professor Sanjeev Sam Gamber. So both these professors gave me opportunity to learn more in this area of molecular imaging in Stanford. He's recently passed away So he's a very good person. And Professor Suhang Lee and Professor Hansu Park, both are my previous PhD supervisors who gave the opportunity to do the PhD in Korea. And this our wonderful team of McCarthy Lab, and his our instructor Dr. Chase. He did most of animal studies here in MMRI. At the end, I want to thank the organizing committee from Kununad Arts and Science College. Thank everyone who's without your contributions I cannot make this seminar possible. Thank you so much.
Thank you very much, Dr. Jagadish Chandra Bose Rajendran. It's really an inspiration for our students, even for us also. Your team is very up to date in the knowledge of till you were just published the contribution in a COVID also. So this is possible when the people have an idea. So this has been more eye-opening for our students studying biochemistry, what we can do, what will be next, like that. So this is one of the inspirational talk for us, for our students, that they can move if they have a zeal to move on. They can move in any level in the biochemistry field. So really, we are happy to have you in this occasion, and I would like to invite Dr. Nirupam, Organizing Secretary and Professor of Biochemistry, to deliver a vote of thanks. Over to Dr. Nirupam.
Good morning to all. Sir, may I audible, sir?
Yes, ma'am. Yes, ma'am.
I feel honored and privileged to get the opportunity to propose a vote of thanks on the webinar series to those who have directly and indirectly contributed to this event in an effective manner. First of all, I thank our Honorable President, Dr. M. Aruchami, sir, who raised us support and guidance. We are extremely thankful to our beloved Secretary, Dr. C.A. Vasaki, Madam, for giving me the opportunity for conducting this program. I also extend my sincere thanks to our principal, Dr. M. Lakshmanaswamy, sir, for his help to conduct this webinar series. I would like to thank our Associate Dean, R&D, Dr. R. Sivaprakash, sir, and Dean Academics, Dr. S.R. Madan Shankar, sir, for the great support to facilitate the gathering. I extend thanks to our today's resource person, Dr. Jagadish Chandra Bose Rajendran, postdoctoral researcher. He summarizes the recent representative research regarding the nanoparticles and biomedical applications of cell-derived vesicles, which represent important research in the focus areas. I thank all the staff members in my department to give such support to making this webinar a success. My special thanks to the HOD, Dr. B. Selvi Madam, for her guidance to and limitless support rendered for conducting this international webinar. Thank you, Madam. My heartfelt thanks to Dr. R.T. Narendra Kannan, Session Coordinator, Assistant Professor, Department of Biochemistry. Thank you, sir, for making this wonderful webinar series in a great. Thank all the participants in our episodic success. On the whole, the webinar was fulfilled with plenty of information. I hope the students and participants might have benefited out of it for his great success. I like to thank all the resource persons for accepting our invitation and effectively shared their knowledge with us. Hope all the participants have enjoyed the sessions and might be useful to your research career. And it is my great pleasure to thank all the participants to join us. I would like to thank all your and your cooperation. Dear participants, those who have submitted the feedback for all the 4 sessions going to get the certificate in a week or 10 days. In this regard, you can contact me for any time. Once again, I thank you for all your attention. Have a good day.
Yeah, thank you, ma'am. Thank you very much. Here, I just wanted to summarize the 4-day session. First day was Dr. Ravi Mardachalam. He is from ISER Trivandrum, talked about hand scrub and puzzle of signal respiration. And the second day program was Dr. K.B. Rebijit on the topic of molecular approaches, insect pest management. He was from New Zealand. On the third day, Dr. Srinivasan Jayaraman from Malaysia. We spoke about the nanoparticles and an alternative antibacterial agent. And today, Dr. Jagadish Chandra Bose from USA is talked about cell-derived vesicles, isolation, functionalization, and biomedical applications. Sir, actually, we are a team of working in nanomedicine also. So hope it will be initiation to join venture to do our research jointly in the future, near future. So we look forward for your help also in this to develop our research the nanoscience in technology. And I request the participants to fill the feedback form, and we will be the same feedback form only for inaugural session also. So you can again, no need to fill up again. It's fine. This is enough. So we will be sending the certificates within a week or 10 days, as ma'am told. So thank you for your valuable time spending with us, and thank you so much. And we look forward for our further programs also. So keep in touch with us, and we'll be looking forward to you also. Thank you so much. Thank you for everyone. Thank you. Have a good day.